Well, good afternoon, good evening, good morning, everyone, and welcome to Facebook Live with the Cutaneous Lymphoma Foundation. I'm Susan Thornton, as many of you know, the CEO of the Cutaneous Lymphoma Foundation, and we're really excited today to have with us our first guest in trying to bring this kind of interaction and interview to all of you at home, sitting in front of your computers. So here we go. We have Dr. Ellen Kim, who is at the University of Pennsylvania and one of our renowned cutaneous lymphoma experts in the field. And we're really delighted to have her with us today to talk a little bit about the recent scientific and medical meeting that happened in London about a week and a half ago that we both attended. And we wanted to be able to bring some of the highlights from that meeting to all of you, so that you can hear a little bit about what happened and what was shared. And Dr. Kim has so graciously agreed to join us today and answer some questions and share with you some of her insights and highlights from the meeting. So thank you, Dr. Kim. It's really great to have you.
Thank you, Susan. I'm so happy to be here and to see the CLF doing these kind of videos in conjunction with the Facebook pages.
That's really great. And I should also mention that Dr. Kim is our new chair of our Medical Advisory Council and has really been engaged with the foundation for a very long time. And we're thrilled to have her with us. And actually, this is the first time we've done this kind of an interview. So hopefully all the technology will work and we'll get rocking and rolling. And before we get started, just a quick announcement to let everyone know that you can post questions, I think, right below the video, and we'll do our best to try to answer them during the course of the time we have. And then also, just to let everyone know that we have a patient education program coming up in Paramus, New Jersey on November the 11th. And you can find out more about that on the website, clfoundation.org. So without further ado, we put together some topics that we thought we'd try to discuss. There's probably more information that we could talk about than we'll have time for, but since we only have a half an hour. But Dr. Kim, we thought we'd review briefly some of the biology insights that were shared at the and what's happening in the arena of clinical trials and some new things coming down the pike. So to get started, how about if we talk a little bit about some of the biology that was discussed at the meeting? I know from my perspective, there's a lot of science and there's a lot of pretty deep information that gets shared, which may not have direct relevance to a patient, but all of this kind of information is really important in understanding the disease better from a clinical and a research perspective, so that we can learn more about the disease and build treatments and therapies and treatment regimens in order to help patients. So I'm going to stop babbling there and turn it over to you. And if you can share a little bit with the folks about some of your insights into what was shared at the meeting.
Certainly. I think everyone is hearing on the news the buzzwords personalized medicine, and that is a general theme that many scientists in CTCL are trying to pursue. So several groups presented their data using newer techniques, sequencing techniques, which are ways to look for mutations in individual cancer cells, and to see if we can find any clues as to why the disease gets worse in some people, and in other cases to see if there are mutations that we can target with current therapies or future therapies. So we're still in the early stages of development for this type of work. It's really exciting because we have the tools to actually look now in individual patient cells. And so they've done it in advanced stage disease, and there was a group from Harvard that looked at it in early stage disease, and hopefully with more studies, we'll see some patterns and things that we can take action on.
Wow, that's really great. And that's a very good insight in a nutshell from a lot of presentations that went on, I think, most of the day on Friday afternoon. And I think that, again, from my perspective, this is really critical so that we can then do a better job, right, as you said, of personalizing the treatments, understanding an individual's disease particulars, and then being able to put a treatment plan together for them that best fits their specific disease biology, if you will, right? So early on, and a lot of really, really great work. So then kind of moving through the way that the meeting was structured, from that, the biology component, we moved kind of into the diagnostics and biomarkers, which from my perspective is kind of the next layer on top of that biology. So once we begin to better understand the biology and the disease and how it acts and the impact on the cells and the environment, then we can start to look at how do we use that diagnostically, what are those biomarkers. I know, a lot of people have heard in other cancers, very targeted markers that can direct therapies and things like that. And we don't quite have one thing. There's probably a number of things, and at least that's what I end up taking away is, wow, there's a lot to this umbrella disease of cutaneous lymphoma, but we're making progress. So any insights that you might have from that arena?
One of the main questions I hear from patients pretty much every visit when I see a new person is that they wonder why it took so long to get diagnosed. And so that's one of the major goals of all of us in the field, is to try to shorten that time so that we can start treatment and relieve patients' symptoms earlier. So there's a newer form of sequencing called high throughput sequencing that is available at some specialized centers. And they're looking at that as a way to make earlier diagnosis and maybe more specific diagnosis. Other like biomarkers that were interesting were biomarkers looking at what they call immune checkpoint receptors. They also looked at things, receptors on other immune cells that help fight the cancer. So like macrophages and other immune cells that help, attack the cancer and get rid of it and eat it, so to speak. So it's kind of exciting to see this new immunotherapy and the immunotherapy biomarkers that are being investigated more in CTCL. They're already being used in things like childhood leukemia and B-cell lymphoma. So many patients, you might have heard the groundbreaking news of engineered T cells being developed, and immunotherapy really curing disease. And all of this, immunology biomarker work is one step on the way towards trying to achieve that in CTCL.
Excellent. And just for some of those folks that might be joining us right now, we are speaking with Dr. Ellen Kim from the University of Pennsylvania, and we're talking about some of the highlights from the recent European meeting that was held in London with, I think, about 350 researchers and clinicians from all over the world, and discussing all about cutaneous lymphoma. And we're talking a little bit now about diagnostics and biomarkers. And I think one of the questions, and this is a little maybe off the mark, but I know in the news and a lot of people have heard about CAR T cells, and that's a big buzzword, not quite ready for prime time, and not, since our disease is a disease of T cells, that probably isn't something that will be relevant. But these other biomarkers and looking at how the immune system functions is something that a lot of folks were talking about at the meeting, right?
That's right. I think that this comes back to the whole issue of also early diagnosis. We don't have one specific tumor, specific marker to make an early diagnosis and to also target specifically to get rid of CTCL. So that's of course our ultimate goal. There is progress towards T-cell lymphomas. There's 2 clinical trials for systemic CD30 positive T-cell lymphoma that are using CAR T technology. I think one at MD Anderson and one in North Carolina. They're not enrolling CTCL patients yet, but we'll await their results once they're published eagerly and see whether or not it's a good risk-benefit ratio for our patients.
Wow, that's great, and that's kind of new news for us, and we'll be making sure to stay on top of that and see what happens. And actually, a couple of notes here. So people were asking about CAR T, our disease, so you just answered that. And Alex, congratulate you on your advisory, your chair of our Medical Advisory Council. And Terry wants to know if there's a higher risk of depression and suicide. Thoughts on targeting, and I don't know if that's related to therapies or, maybe there's some interactions with some of these newer treatments, which may come into play, and we probably don't know enough about that yet, would be my sense. But, targetin. Okay. Terry wants to know if there's a higher risk of depression or suicidal thoughts if you're on targetin. A little off the mark, but I don't know if you have any.
Well, so the short answer is targetin, no. But targetin is what they call a retinoid. So it's a vitamin A-derived medication. And it is related to some other pills that you might have heard of for other skin conditions. So Accutane for acne, Soriatane for psoriasis. And, there have been a lot of studies looking at whether or not those groups of medicines can affect mood and depression. And I think the consensus is that depression and suicide are not increased with this group of medicines, not based on really strong retrospective studies in large groups of patients. However, can they affect mood? They might in some patients. They might. And so it is important to be monitored and to notify your doctor if you're experiencing some mood changes on targetin.
Great. Thanks. That helps. And I know we actually have a hard stop, so we've got about 15 minutes left to go. I thought we could talk a little more about the therapies that were discussed, and I think for me, exciting to see all the activity in clinical trials, both a couple of trials that have completed where we're starting to see data being published with the hopefulness that they'll become approved treatments here in the not too distant future. And then some of the new things that are either just coming out of phase 1 clinical trial, going into the next phase 2, and then some that are just in the early stages, clinical trial stages. But to me, in a rare disease where most rare diseases have no treatments, the fact that we've got what seems to be an explosion of activity and research going on and some new treatments that are in the clinical trial process. I don't know where you'd like to start on that, but maybe talk a little bit about the two treatments that are kind of have wound down their clinical trials that I know one would be obviously brentuximab vedotin. A lot of people have heard about the Alcanza trial, and I know we talked about that as well. Maybe we start there, just if there's anything exciting, which I think.
So folks might have noticed that on my disclosure slide, I list several companies that I work with. And so these multicenter clinical trials, essentially all the major cutaneous lymphoma centers, we have to collaborate and work together to be able to offer these treatments to patients. And then if through the generosity of our patients volunteering, and these wonderful, sponsors who are willing to develop the medication, hopefully some of these can come to market and help people. So the Alcanza trial was one of the largest in CTCL history, and we're really proud of it because it enrolled 131 patients, and it was what we call a randomized control trial, which in the medical world is the most objective way to see whether a treatment works. And this medicine is an antibody. It's called a monoclonal antibody drug conjugate, and it is combined with a special medicine that basically helps kill the T cells, and it targets a receptor called CD30, which is present on some cutaneous T cell lymphoma cells, particularly in patients with more active or aggressive disease. And so this medicine is called brentuximab vedotin, or Adcetris is the brand name. And it's been around for a couple years because it's been FDA approved for Hodgkin's lymphoma and something called systemic anaplastic large cell lymphoma, which is an internal T cell lymphoma that expresses CD30. So this trial was completed, and the results were published in Lancet, a very prestigious journal, and it demonstrated that this brentuximab, which is given IV once every 3 weeks, was definitely more effective in treating CTCL patients who have stubborn disease compared to the control arm. And the control arm was either methotrexate or bexarotene, and the results were pretty impressive. So they presented that data, and the safety profile seemed pretty favorable. The main side effect we have to monitor for is what we call neuropathy, or numbness and tingling in the sensory nerves. So that was it's a pretty impressive trial, and we'll be using it more. We'll be using it more, particularly in patients with plaque and tumor disease that's not getting better with the skin therapies and the other therapies that we have currently on the market.
Awesome. So actually we have a question. Mike is asking, can you check for CD30 counts in a flow cytometry test to determine if you're a good candidate for the treatment? I think that's a great question.
So for skin biopsy, there are a couple ways you can check for CD30. On skin biopsies, the pathologist can do something called an immunohistochemistry stain and see whether the CTCL cells express CD30. And that's done pretty routinely in many specialized CTCL-oriented centers. But not always for early stage disease. And flow cytometry is a specialized test that uses antibodies as well, but it's primarily done on the blood or lymph nodes. So that is one tool to use to look for CD30. Most centers are not, though, using it on skin biopsy specimens because you really need a large sample, a large piece of tissue. So most labs are using just standard staining for CD30.
Awesome. And I was going to kind of set up the next, well, actually there's a couple of other things, but someone asked about any news on Mobileuzum AB. So that was also discussed at the meeting and finished up the clinical trial last year. And I know there's not a lot we can talk about yet because things haven't quite been published, but.
Well, it's wonderful to see how up to date all of our audience is, and they're keeping track of these new therapies. So I would say stay tuned. Right now, we participated in the trial. It was also a really large multi-center trial, and they're going to present the abstract at the American Society of Hematology meeting in December. We're kind of optimistic about the results, but the official results will be published then.
Right. And they're working hard on their submission to the FDA for approval. So we'll let y'all know, and after ASH, we'll have an update, another Facebook Live actually after the hematology meeting, so we'll hopefully be able to share some more then. And then you presented actually on Valcor and some of the projects you've been working on.
Yes,
well.
It's an old medicine that has a new form, and it is it's now in Europe. So that was the big news as of this year. It was approved in the U.S. several years ago. It goes by there are a couple names: nitrogen mustard gel, or mechlorethamine gel are the older names, and the brand name is Valchlor gel. And in Europe, the formulation is chlormethine and methamine, and it's called Ladaga. So I think the important thing to know about this medicine, it's not a new medicine. It's been around for a long time. It's been a mainstay of early stage disease treatment. It has good efficacy and good safety profile. But now it's FDA approved in the US. So the coverage regarding insurance coverage is a bit easier than when it used to be just a compounded ointment or in solution form. So we presented data. We were kind of interested in looking at how Valchlor gel is used in the real world. It's one thing to see the results from a clinical trial, but we were curious to see, well, who's using it? Is it just early stage, or are we seeing late stage patients use it, as adjunctive therapy? What's the side effect profile? Are people combining it with steroids to minimize side effects? So this was a registry trial, and thank you to all the patients who agreed to participate. And so far, in the 200 patients that have been enrolled, or 300 patients actually now fully, we can see that it is being actively used. A good percentage of patients are using it in combination with other therapies. And the side effect profile, fortunately, is similar or perhaps a little less. So the main side effect is contact dermatitis, which can occur usually within the first 6 months. And it's not dangerous, but annoying. But so far, at least in this observational study, where it's being used in real life, the dermatitis is not worse than the clinical trial, which was reported at about 20%. It's actually maybe perhaps a little less, possibly related to the combination use with steroids, but we need to have further follow-up.
Yeah. Which I think is a great, for folks to appreciate that even though something may end up getting approval and is available, there's still a lot of other work in clinical trials that get done looking at a lot of different either ways of using it and combinations, especially in our disease. I know we only have about 5 minutes, so we have a couple more questions if you don't mind. We'll see if we can get these answered. Someone asks if brentuximab is effective for Sézary.
In the trials that were published, there were Sézary patients, and they did see effect. One of the issues is that it's not every flow cytometry lab has CD30 to look at the expression on Sézary cells. And the numbers of Sézary patients treated in the 3 trials, this current trial and the previous 2 phase 2 trials, they're not high numbers. So the bottom line is we don't know. It's worth trying if your doctor thinks it's advisable. But the best data right now is in plaque and tumor MF. And then I forgot to mention, you may hear from your doctors. We are actually, several authors have published data, and they discuss this, that CD30 expression is really variable in the CTCL lesions. So sometimes your doctors might want to take more than one biopsy to see if there's CD30, because sometimes it can really vary. And so because of that, they are trying it even in patients who don't have necessarily high CD30 expression, because they saw responses in patients where even the CD30 expression was on the lower side. So that's, I think, interesting, and we need to, we'll have more follow-up later to see if that bears out to be true in real clinical practice.
Yeah. And another reason for ongoing investigations, even after drugs get approved, to see how they may affect, as you said, in the real world, right? So another question, see, these folks are so smart, asking what about the IPH4102 trial, which is just coming out of phase 1. And maybe we only have a couple minutes, so maybe you can talk a little bit about what happens in phase 1, and then, we got some good results and now they're going into phase 2, and maybe a little bit about this targeted therapy.
So that trial and that medicine particularly targets a marker on CTCL cells, and seems to be better for Sézary syndrome or erythrodermic patients with blood involvement. A phase 1 trial, you don't hear a lot about it in the news and the lay press because the purpose of a phase 1 trial is to determine safety and the optimal dose. And they don't really closely look at whether or not it works quite yet. It's not designed that way. So they really look at different doses and they see what the maximum tolerated dose is. They look at safety profile and see whether they can further develop it into a phase 2 or phase 3 trial, which is where they look at efficacy or clinical effect. But the good news is that that trial showed a good safety profile, and they optimized the dose. And we look forward to seeing the results once they expand it into a phase 2, phase 3.
Wow, that's great. Okay, so now we're sort of down to the 2-minute warning, and I know you have a hard stop at 6 o'clock Eastern time. Just to let everybody know, what I thought was extremely exciting about this meeting was there were presentations on quality of life specific and some presentations about the impact on caregivers. And we'll talk a lot more about that next month with Sue McCann, who is one of the nurses from the University of Pittsburgh, who also joined us for the meeting and the first ever international Nurses Network meeting that happened as part of this overall program. Let's see, I'm just trying to think. Anything else that you think was a highlight that folks might want to know? Again, there's probably a ton of things. It was a pretty dense and packed meeting with a lot of presentations. But if there's anything else you'd like to share before we let you go.
Well, I would just applaud and encourage all patients out there who donate their time and willingness to either participate in therapy clinical trials or observational clinical trials, or even agreeing to, have their personal story or their medical, what we call tissue banking of specimens to help further cutaneous T-cell lymphoma research and understanding. Everything you just heard today was as a result of patients being willing to fight, to help cure this disease. And so please continue that. We're all grateful that we have such a great community of patients who are devoted to improving things.
Well, thank you. Well, and I think that goes back the other way too. I can share with everybody, I have the privilege of attending and getting to know so many of the researchers and clinicians around the world, and I can really say it's an amazing community of dedicated individuals who really truly have spent their careers and are spending every day working on our behalf to try to figure out the disease, to better ways to treat the disease. And that's Dr. Kim's messenger saying, it's time to let her go. But anyway, thank you, Dr. Kim, for joining us. It's really an honor and a privilege to have you. It's great that you took the time to be able to share a little bit of the highlights with everybody live. And we'll be doing these interviews once a month. If you have topics and things that are of interest to you, let us know, and we will pull in the experts and the researchers and have a conversation. And thanks again, Dr. Kim, and we look forward to seeing you maybe again here on our Facebook Live interviews, and certainly engaged in all of our activities over the next years to come. So thanks so much and have a really great night.
Thank you. Thanks.
All right. Have a good one.